So hello everyone, and welcome to the first guest speaker event at Medway School of Pharmacy. So our today's speaker is ParthivePatel from the University of Bristol. So Parthiv started his degree in chemistry at the University of Chicago and then went on for his PhD in molecular biology at the University of California in Los Angeles. After that, he joined the lab of Bruce Ecker with fellowship from the American Cancer Society, initially in Seattle, but then the lab moved to Heidelberg at the Cancer Research Center and the Center for Molecular Biology in Heidelberg. So since 2019, Parthiv is in UK, where he studied his own lab with a fellowship from Elizabeth Blackwell Institute. And since 2020, he is a Sir Henry Dale Fellow from the Wellcome Trust and the Royal Society. So Parthiv, thank you very much for accepting the invitation. We're very happy to have you here. We're looking forward to your talk.
Yeah, you're welcome. Thank you, Fanny, for that nice introduction. And I want to start by thanking the Medway School of Pharmacy, the University of Greenwich and Kent for inviting me to speak today about our work. So I just started my lab at the University of Bristol almost 2 years ago. And I'm going to talk to you about some of the work I did as a postdoc in Bruce Edgar's lab in Heidelberg, and how that is leading up to the work that we're doing at the University of Bristol. And I'll go over some of the projects that we're also doing there. Okay, so the central question in our lab is, how do tissues maintain themselves under stress? So many of our epithelia that are exposed to the environment, like our lungs or our gut, they lose cells under a constant basis. And stem cells often divide and replace those lost cells. And this maintains the tissue over time. Now, occasionally there'll be acute damage and many epithelial cells will be lost. And so the stem cells need to then rapidly divide in order to replace all those lost cells. And these two processes of maintaining the tissue and regenerate the tissue maintains this tissue over the lifespan of your organism. Now, if this process goes wrong, this can lead to tissue dysfunction, degeneration, as well as aging of the tissue. So an important question is, how do tissues understand that there has been stress, and how do they relay to the stem cells in order to make them rapidly proliferate and replace those lost epithelial cells? So today, I'm going to talk to you about three things. First, generally, I'm going to talk to you about the process of tissue maintenance and also how stem cells, in my case, intestinal stem cells, maintain a tissue over time. Secondly, I'm going to tell you a story about how the intestine or epithelia sense damage within the tissue and how it promotes regeneration, in this sense, intestinal regeneration. And then I'm going to end by an older story whereby stem cell-derived tumors can induce a niche, which is actually very reminiscent of a regenerative niche, and how it can subvert it for its own purposes and growth. So as I mentioned, the process of tissue homeostasis is the process where the number of cells that are gained in a tissue is equal to the number of cells that are lost. And this is often kept by an unspecialized cell called the stem cell, which divides and gives rise to progenitors that differentiate, that incorporate into the epithelium. When the number of cells that incorporate into the epithelium is equal to the number of cells that are lost in the epithelium, that we consider tissue homeostasis. And the stem cell can reside in a tissue for a very long time because it has a special property of self-renewal. Now, this process can go wrong, as you know. For example, the stem cell can be lost over time, or it could it could proliferate less and make less progeny. And this could lead to more cells that are lost in the tissue than there are gained. Thus, this could lead to tissue dysfunction and aging. In contrast, you could have the opposite situation where the stem cell proliferates too much or fails to differentiate. And this leads to an accumulation of cells within the tissue that is greater than the number of cells that are lost. And this could lead to tumorigenesis. So the proper balance between the number of cells and loss is very important to maintain tissue homeostasis. So to understand this question, we are using this very simple model called the adult fly midgut. It's pictured here in red in this cartoon. It's the stomach and small intestine of the fruit fly. And here's a confocal image of an adult fly intestine. It's a long tubular organ, not very different than ours, right? It looks like a long intestinal tube. And it's a very valuable model for the last 14 years to understand how many processes of tissue biology, for example, stem cells and how they maintain stemness, how they differentiate, tissue renewal and regeneration, which my lab concentrates on, aging, tumorigenesis, as well as host pathogen interactions, and many, many more questions. So the fly intestine, you would think, okay, it's very different, it's a fly, but actually has so many similarities to our gut. For example, it's maintained by intestinal stem cells that divide and give rise to two mature cell types, the nutrient absorptive enterocytes, or the hormone-producing enteroendocrine cells. And these are the same cell types that are found in the mammalian intestine. So this is a cartoon of a mammalian intestine here. And here in the mammalian intestine, the stem cells reside in this pocket-like structure called a crypt, where the stem cells divide, produce progenitor cells, which are brown here, and they differentiate into enterocytes or enteroendocrine cells, the same exact cell types you find in the fly. And interestingly, the stem cells use many of the same signaling that occurs between both systems. For example, how stem cells self-renew themselves, how their proliferation is regulated, or even their differentiation is shared between mammals and flies. So this makes the fly intestine a good model to use to understand some mammalian biology as well. So the lineage is pictured here. So the stem cell, as I mentioned, maintains the tissue over time. It divides and produces two main types of progenitor cells, an enteroblast, which differentiates into an enterocyte. The enterocytes are the cells that actually aid digestion and absorb the nutrients. The enteroendocrine precursor then differentiates into an enteroendocrine cell, which makes hormones, which facilitates processes such as peristalsis, or the movement of the bolus along the tube. And all of these cell types can be identified using various markers. Now, one thing I should mention before I talk about that is that the stem cell uses NOT signaling between the enteroblast, the one that's destined to become an enterocyte, and itself to differentiate this enteroblast into an enterocyte. Therefore, the intestinal stem cell can often be identified by the expression of the Notch ligand delta in flies. You can see that here in red. And this green signal is the expression of a transcriptional regulator called escargot, which is expressed in the progenitor cell population. Now, around these progenitor cells, you have these large polyploid cells, which are the large enterocytes that absorb nutrients, and they can also be identified by other markers such as PDM1. And the enteroendocrine cells can be identified by their expression of a transcriptional factor called prospero. So all these cell types can be identified, which makes it easy for us to quantitate the number of cells that are around in that in different conditions. So when I first started my postdoc, I had first two questions was, well, are these stem cells important for maintaining the tissue? It was assumed that it would be. So what I did is I tested if you ablate the stem cells, what happens to the tissue? So the green cells that you see here are the progenitor cells. And if you overexpress something that causes them to be lost over time, you can see that the tissue shrinks and all the stem cells are lost from this tissue, as well as many of the mature cell types. So the stem cells are important for maintaining the tissue over time. Another question I had is, would these stem cells become activated if mature cell types are lost from the tissue? So to do that, I devised an experiment where I ablated the mature enterocytes from this tissue, which make up 90% of the epithelium, and looked to see whether the stem cells start to divide. Now, under a control midguts here, you see a control situation. Midguts don't have many phosphorylated histone 3 positive cells. This is a mitosis marker. And majority of cell divisions are by stem cells. So you can see that most of the stem cells are not dividing in this normal unchallenged condition. But if you ablate the enterocytes, you start to see these phosphorylated histone 3 positive cells. And you can have this graph here with the number of mitoses per intestine or midgut over time after ablation increases. And if you stop this ablation signal, you see that the stem cells start to divide, continue to divide, but they will eventually stop over time when the tissue reaches its final size again, and eventually proper cell number. So these data suggest that stem cells are important for maintaining the tissue in the fly gut, and stem cells are important for regenerating the tissue upon acute cell loss. Now, for many years, I and many groups have been trying to understand, well, how does a stem cell know it needs to divide when you lose these mature cell types? And so we try to identify what is this fly midgut regenerative niche, okay? And regenerative niche is quite complicated. I'm going to try to give you a very simple view, but to understand that, researchers have been trying to expose the midgut to many types of stress and then identifying pathways that become activated. So types of stress that we use, one very common one is pathogens that are specific to flies, such as ECC15 or PE Pseudomonas entomophila, which is what I'll show you a lot in my talk. Reactive oxygen species that cause damage, such as hydrogen peroxide, or generators of reactive oxygen species like paraquat, detergents like SDS or DSS, DNA damage with bleomycin treatment. I found that tumor growth can also stress epithelial cells, and also cell detachment. Now, one of the signals that are very prominently activated upon damage is the Jun N-terminal kinase pathway, or JNK. And this pathway actually turns on cytokine or IL-6-like molecule, interleukin-like molecule called unpaireds. These unpaireds stimulate stem cell proliferation by causing the cytokines binding to the domeless receptor and activating JAK-STAT activity in the stem cells, which drives their proliferation. And this JAK-STAT activity is also important for the daughter cell to differentiate into these mature cell types. Now, this is not the only pathway. There are many pathways that become activated. And one important pathway I just want to mention is the EGFs, which are produced by many other cell types within this tissue. And they also activate proliferation of the stem cell by through this EGFR RAS-MAP kinase signaling. Okay, so at this time, we knew a whole bunch of signaling pathways that become activated upon damage, but we hadn't known whether another stress-activated protein kinase pathway, P38, was involved in this process. So I went out to look for that during my postdoc, whether it was involved in sensing damage and then promoting regeneration, possibly through unpaireds. And we also knew at that time that reactive oxygen species from an NADPH oxidase called DUOX, or dual oxidase, was important upon damage to stimulate stem cell proliferation after bacterial infection as well. And what we had assumed as a community for many years is that DUOX produces reactive oxygen species which damage the gut and activate JNK, and this causes them to produce unpaireds, these enterocytes to produce unpaireds and stimulate stem cell proliferation, okay? So that was the mechanism that we assumed was happening. Now, I also became interested in another NADPH oxidase called NOX, because it was involved in producing reactive oxygen species upon the encountering of commensal bacteria. And so there was no role that was involved with regeneration for NOX. So I also wanted to test whether NOX was important. And NOX and DUOX have been shown in other species to be very important for regeneration, for example, in the zebrafish fin regeneration. So as you can see, it's a very complex process with many pathways becoming activated. And also other cell types are recruited to this gut, such as hemocytes, which are like macrophages that come and actually also participate in this process. And later, it was discovered that even tracheal expansion occurs, which is kind of a vascular system for Drosophila to support regeneration and provide oxygen. All right, so the next part, I'm going to talk to you about how we then try to look at P38 and these NADPH oxidases and identify how how damage is sensed within the gut using those enzymes, and how intestinal regeneration is promoted in the fly gut. So, but before I do that, I just want to go over the stress-activated protein kinase signaling pathway. This is a very important part of this second part. So in flies, there are two branches, the Juna and terminal kinase pathway, or J&K. There's only one J&K in Drosophila. There's also the P38 branch, and there are three P38s in Drosophila, but I'm only going to talk about P38A and B today because they're the only ones that can be regulated by phosphorylation by MKK3, or in Drosophila, it's called licorn, or the unicorn. And this SAP2 kinase can be regulated by SAP3 kinases, or stress-activated protein kinases. And there are 6 of them. I've listed 5 here, but I'm only going to talk about ASK1 and MEKK1 and in their relation to this pathway. So I've highlighted here blue ASK1, MKK3, and P38 because that's the central pathway I will talk about in this part of the talk. So to test whether P38 was a sensor of damage, I used an antibody towards activated P38 or phosphorylated P38 and infected the flies with this oral pathogen. And normally you don't see active P38 within the tissue, but if you infect the flies, you see an activation of this P38 within this epithelial cells here. So this suggested that damage can activate P38 in this epithelial cells. So what I showed you that if you ablate the mature cell types, stem cells can divide, right? So if you give this oral pathogen, you see stem cells start dividing in the controlled midguts compared to unchallenged controlled midguts. However, if you remove P38A and B from these enterocytes and then infect the flies, the stem cells don't divide as much. So this suggests that P38 senses stress in these enterocytes and then somehow non-autonomously stimulates stem cell proliferation. Okay, so then to check whether licorn was involved, we knocked down MKK3 by RNAi and infected the flies with Pseudomonas entomophila, PE. And you can see that there is less active P38 compared to in control infected midguts. So pathogenic infection can activate P38 via MKK3, and this could lead to stem cell proliferation. And we showed next that if you just simply overexpress MKK3, you see you can activate P38, but also you can overexpress licorn and see you can get a lot of stem cells to divide as well. So this data together with the previous slide suggests that P38 senses stress through MKK3, and that MKK3 P38 signaling can drive stem cell proliferation in a non-autonomous way. But how does it do that? And as I mentioned, the cytokine unpair 3 is very important for stem cell proliferation, and is usually induced upon damage. But we found that enterocytes where P38 is depleted, unpairs are only upregulated by 50% of what they normally would be. So this signal is reduced, suggesting there is less JAK-STAT activity in those stem cells, and thus they're dividing less. To test that, we looked at the STAT activity reporter. And you can see normally under unchallenged situations, STAT activity is observed in the progenitor cells. But if you infect with PE, you see STAT activity throughout the midgut. But if you remove P38A and B from the enterocytes and then infect the flies with PE, you see there's a lower level of STAT activity in the midgut. So this data suggests that the enterocytes produce unpairs via P38 signaling, and this leads to an inhibition of STAT activity in the stem cells, leading to a failure in intestinal stem cell proliferation and regeneration. So that was interesting because previously it was shown, for many years, we thought that P38 becomes activated upon pathogenic bacteria because of recognition of these pathogens. So a group had studied for a long time how pathogens are recognized and can activate P38 through this process. So, but what I want to stress here is that P38 may do that, but it does definitely do that due to damage to the epithelium. And that's because we can damage it with a variety of types of damages, and it all activates P38, regardless if there are pathogens present or not. So for example, reactive oxygen species can activate P38, such as hydrogen peroxide. Detergents can activate P38. So here you can see SDS feeding of flies, oral feeding of the SDS can lead to activation of P38 compared to control flies that were fed water alone. Tumor growth can activate P38. So cells that are surrounding the tumor can activate it. Cell detachment can activate P38. And you can see here that if you knock down an important beta subunit of the integrin complex in the midgut, beta PS1, you see there's an upregulation of P38 within these detaching cells compared to in control. And also physical wounding with a tungsten needle can activate P38 in the epithelial cells. So this suggests that actually P38 could be activated not only by the presence of bacteria, but also through the damage that other types of stress may cause. I'd like to just first introduce what we thought about P38, and then I will go into the mechanism as to how P38 becomes activated upon damage. So as I mentioned, P38 was thought to become activated by pathogenic bacteria via the upstream kinase MEKK1, which is a SAP3 kinase. And P38 activity was thought to upregulate dual oxidase or duox via ATF2 activity. And this conferred to the gut reactive oxygen species mediated intestinal immunity. So if you have a pathogenic infection, these reactive oxygen species fought off this pathogenic bacteria infection. But this was due to the fact of an activation of P38 via the recognition of pathogenic bacteria through peptidoglycan sensing. Okay. Now, reactive oxygen species were also produced by another NADPH oxidase, NOx, against commensal bacteria. And it's thought that this enzyme was important for maintaining the amount of commensal bacteria you have. And without it, you get a dysbiosis of commensal bacteria. Okay, so this would suggest that upon pathogenic entry, bacterial entry, this would activate the expression of duox, lead to ROS production, activate a signaling pathway such as J and K due to damage, and lead to regeneration. But that is not what we saw. What we saw instead was that P38 can be activated not only by pathogens, but by other types of stress. This can lead to stem cell proliferation and intestinal regeneration. And we know that intestinal pathogens can cause ROS production to occur. But what we found is that a ROS-sensitive kinase, ASK1, was involved in this activation of P38 and not MEKK1, as we would predict from this older model. So to show you this data, here you can see that if you deplete ASK1, which is really a ROS-sensitive kinase, it actually reactive oxygen species change the oxidative state of cysteines within this protein. If you remove ASK1 and infect the flies with PE, you see it does not activate P38 compared to in controlled midguts. So pathogenic infection can activate P38 by ASK1, and this could lead to stem cell proliferation. So to test whether ASK1 was required for regeneration, we knocked down ASK1 in the enterocytes, infected the flies with PE, and you can see there's a reduction in stem cell proliferation compared to in controlled midguts. So upon pathogenic infection, P38 activation and stem cell proliferation required ASK1. Now, interestingly, we found that MEKK1 was not required for P38 activation, unlike was proposed before, nor for intestinal regeneration, even if we looked at whole body mutants, or even if we knocked down MEKK1 within the enterocytes themselves. Now, next, wanted to check whether P38 activation can be, other types of damage can also go through ASK1, for example, ROS or detergent stress. So we expose the flies to hydrogen peroxide, so oral feeding of hydrogen peroxide leads to active P38. But if you inhibit ASK1 within the epithelium and then feed the flies hydrogen peroxide, you see that P38 is not activated in this gut compared to in control. And we also see the same with detergents. So detergents can activate P38, but if you remove ASK and give detergents, you don't see an activation of P38. So this activation of P38 due to other stresses like ROS or detergents also required ASK1, and this led to stem cell proliferation. So to answer whether it is specifically reactive oxygen species, we try to reduce the amount of reactive oxygen species in those enterocytes by overexpressing enzymes that would clear reactive oxygen species like superoxide dismutase SOD1 or catalase. Both of these convert superoxide and hydrogen peroxide to water and oxygens, thus neutralizing the ROS within the cell. And then we also overexpress NRF2, which is a master oxidative stress response transcription factor. And what we found when we used these treatments is that it reduced the amount of active P38 upon infection compared to control Midguts. So that data also suggested that ROS can activate P38 as well. And is that important for stem cell proliferation? We did the similar type of experiments as previously. We overexpressed NRF2 and then infected the flies and found that there were fewer stem cells dividing in those midguts compared to control midguts. And with detergents, you see the same thing where you overexpress NRF2 and you provide detergents such as SDS, and you see less stem cell proliferation in those midguts compared to in control midguts. So together, these data suggest that infection or detergents can activate P38 via reactive oxygen species, and this can lead to stem cell proliferation and regeneration. Now, but what was the cellular source, right? So we knew that ROS was required, but what is producing these reactive oxygen species? And I'll go back to these NADPH oxidases like NOx and DUOX. You see that if you knock down NOx and infect the flies with PE, you see there's less active P38 in this epithelial cells compared to in control. So this suggests that pathogenic infection can activate P38 via ROS that's produced by NADPH oxidases. But interestingly, DUOX had no role. So both NADPH oxidases, NOx and DUOX, both make reactive oxygen species, but somehow one of them can activate P38 and the other one does not after infection. So then we tested whether NOx was required for intestinal regeneration. And so we knocked down NOx and then we infected the flies with PE. And you see that there is a reduction in the number of stem cells dividing compared to a control. And the same thing with detergent stress. If you remove NOx and provide the flies with SDS, there are fewer stem cells dividing in this gut compared to in control midguts. So this suggests that infection stress or detergent stress can activate P38 via NOx-produced reactive oxygen species, and this leads to intestinal regeneration. So although P38 was not activated by DUOX under infection, DUOX is required for intestinal regeneration. And recently it's been shown that part of that could be from the expansion of trachea that is providing oxygen to the tissue for regeneration. So to summarize this part, we showed that pathogenic bacteria and chemicals such as detergents or physical mechanical damages like from wounding can activate NOx. This produces reactive oxygen species and activates this ASK-P38 signaling pathway. And in these enterocytes, this leads to the production of cytokines. And these cytokines can activate the JAK-STAT signaling pathway in the stem cells, activating their proliferative mechanisms and leading to intestinal regeneration. We also showed that cell detachment and tumor growth can also activate P38, but we still need to test whether NADPH oxidases are involved in that process of activating P38 under those situations. Okay, so I just want to, before I end that part, I just want to go back to this whole model of intestinal immunity that was assumed to also play a role in possible intestinal regeneration. So it was thought that pathogenic bacteria can activate P38 via MEKK1, and this activates through ATF2 dual oxidase expression, which produces reactive oxygen species and then activates the regenerative mechanisms that we already knew. But I showed today that pathogenic bacteria and other stresses can produce reactive oxygen species by activating NADPH oxidases. And this leads to P38 activation via ASK1. We showed that, and this leads to intestinal regeneration. But I also showed you that MEKK1 wasn't required for intestinal regeneration. And also, I didn't get to show you this, but ATF2 was not required for intestinal regeneration as well. So while these mechanisms could still start off regeneration early after pathogenic infection, they are not required for sustaining regeneration over time, unlike this pathway. So now we want to take this information that I learned in my postdoc and try to ask more questions in my lab about how reactive oxygen species promote regeneration other than NOx activating P38, and from more recent literature, how DUOX can maybe activate tracheal expansion. So to do that, my postdoc, Rebecca Wafer, and several students in the lab have been using a three-pronged approach, a candidate approach, transcriptomics, and proteomics to kind of address this. So a candidate approach is that we try to look at whether NADPH oxidase reactive oxygen species are important for the activation of different pathways that are known to be involved in midgut regeneration. We also use transcriptomics, for example, to find out how do stress midguts respond to these stresses, and what part of that is dependent on reactive oxygen species. Is this part of it? Is it all of it? We're also using proteomics to identify proteins that are oxidized upon damage, and then to ask, are those oxidized or reduced proteins upon damage important for intestinal regeneration through genetics? So here's an example of something we would do. For example, we know that NOx and DUOX produce reactive oxygen species. We know that NOx's reactive oxygen species can activate P38, but we don't know what else NOx does. How does it interact with other pathways? DUOX, we know from recent work, is involved in tracheal expansion, but we also don't know what else it's doing in the cell. So this is such a type of question that we're asking and testing various pathways that we know of in the fly gut. We're also then trying to understand, well, how do the midguts respond transcriptionally to stress? We do this by doing whole midgut transcriptomics, for example, from stressed intestines. So we take stressed guts, we isolate RNA from them, perform mRNA-seq, and compare them to unstressed guts. And then we eventually want to remove NOx and DUOX and see what part of that transcriptome changes or is dependent on these reactive oxygen species from these enzymes. Another thing we became interested in more recently was how different midgut cell types respond to stress. And we know a lot about enterocytes, but we don't know so much about other cell types. So we were just trying to devise how to do that in our lab. We've been dissecting guts, dissociating them, sorting out specific cell types, isolating mRNA, and then performing mRNA-seq. So, and lastly, we're using this oxidative, we're looking at redox proteomics, using redox proteomics to look at the oxidative states or reductive states of different cysteines on all the midgut proteins that we can acquire through the process. So we do this by isolating midgut proteins. We label them with one tandem mass tag. First, the reduced cysteines with one mass tag. Then we reduce all the oxidized cysteines, and then we label the reduced cysteines then with a second mass tag. We trypsinize these proteins, take these peptides, and then enrich them, and then send them to LCMS. So in the third part, I'd like to finish by just talking about an older story that before this tissue sensing that's related to intestinal regeneration. And this is because intestinal tumors have, and you'll see, have a lot of, they actually behave almost like wounds, and they create a regenerative niche around them, at which they will subvert for their own growth. So it's well known from a lot of fly literature, even mammalian literature, that the interaction between a tumor and its epithelial cells around it is an important interaction for tumor growth. And so I wanted to study that when I started my postdoc by making first a model to model tumors growing within an epithelium in which you can control, right? So what I did is I used the loss of Notch. So remember from the very beginning of my talk, Notch is very important for the differentiation of the enteroblast into an enterocyte. And if you fail to do this, you lead to tumors that form because you get a failure in differentiation, and then this just forms a tumor. And I'll show you that in a second. But first, I just want to show you these markers. So this is escargot positive cells. These are the progenitor cells, the stem cell, and the enteroblast. And these are enteroendocrine cells that are Prospero positive. And they are surrounded by these large polyploid enterocytes, which are these large cells that absorb nutrients. And as I mentioned, Notch signaling is important between these two cells to promote enteroblast differentiation. But if you lose Notch signaling, this forms a tumor that is a mass of progenitor-like cells that are proliferative and non-mitotic or post-mitotic more differentiated like cells, which are more EE-like cells in this case. And you can see that here in these images, that this tumor mass that's proliferative is positive for escargot, the marker for the progenitors, is also positive highly for delta, which is a marker for stem cells. And these cells are mitotic, so you can only find dividing cells mostly in these cells. Now, the cells that are also part of this mass are post-mitotic, and they're secretory-like, put together they form this tumor mass that grows. Now, one of the first things I identified is that, well, as these tumors grew, you found dead cells surrounding them. You found these pycnotic nuclei. And you can see that this in these EMs. So here's a normal midgut where I've taken an EM image, where this is the enterocyte that is against this visceral muscle that lines the epithelium. And the basement membrane is between those two. And here you can see the lumen with all the luminal contents and this matrix called the peristrophic matrix, which prevents things from coming in from the lumen. Now, if the tumor, when the tumor forms, it forms a mass that actually piles up on top of each other. You can see the cells are all piled up. They're smaller, they're undifferentiated, and they're piled up in a mass. And they take over this basement membrane space and displace these enterocytes. You can see that the enterocyte is very sickly, is depolarized or unpolarized. You see these vacuoles are no longer basal, but they're more all over the cell. And the microvilli look sickly compared to these control ones. So this is likely an apoptotic enterocyte. Now, I wanted to show whether this apoptosis was important for tumors to grow. So to do that, I devised a system where I can make tumors and then manipulate the normal cells that are surrounding the tumor genetically. So if you just make tumors and you don't manipulate the cells genetically surrounding them, this is the tumor that grows out after a week, okay? But if you start the tumors and then manipulate the cells surrounding that tumor and block apoptosis by overexpressing this protein called P35, you see that tumors don't grow as much in this situation. So you need the cell death in order to clear away and make that space for the tumor to expand. So blocking apoptosis in these enterocytes is important for tumor growth. Now, another thing I saw is that many of those signals that I told you that are important for regeneration that are activated upon damage, J and K, or deactivation of Hippo signaling, you see surrounding a tumor. So as the tumor grows, which is this green mass, you see in the immediate cells that are around it, which are normal, J and K activation. You also see Hippo activation or Hippo deactivation or Yorkie activity using expanded like Z. For the fly people, in these cells that are surrounding the tumor, and you can see it's going out further than this tumor mass. And both of these are, both these events occur also in damaged tissues, right? And I told you that these damaged tissues activate the production of unpairs. But you can see these unpairs, using this unpaired reporter, are also produced in cells that are adjacent to these tumors as well. So this data suggested that as the tumor forms and grows, it stresses its normal neighbors, and this leads to a production of unpaireds around this tumor. But the question is this important? It doesn't, but is it important for tumors? Yes. So what I did is I manipulated the cells that are surrounding the tumor for these pathways. So here you can see tumors that are growing on their normal midgut grow out to a certain size. Let's say here 40 cells. But if you remove J and K surrounding those cells, now the tumors don't grow as well over a week. If you activate J and K, you can see the tumors grow even larger. Same thing with the Yorkie activity. If you decrease Yorkie activity by expressing warts or lats in mammals, or you decrease Yorkie by RNAi in those epithelial cells that are surrounding the tumor that are normal, you see they don't grow as well compared to control. And finally, if you remove this downstream target, unpaired 3, also unpaired 2, around those growing tumors, you see that those tumors don't grow as well in these conditions. So these data suggest that tumors not only stress their normal neighbors, and those normal neighbors produce unpaireds, but the tumor actually uses those unpaireds in order to proliferate and grow. Now you can see this unpairs being produced by cells that are adjacent to the tumor and also apical to the tumor. They detach these cells, these tumors detach these cells. You can see that unpairs are expressed in these detached cells. So if you quantify it, like which cells make unpairs or which ones make activate J&K or activate Yorkie, you see this pattern here, where Yorkie activity is activated in fully detached cells, partially detached cells, that would be around here, and also fully attached cells farther away. And if you look at that image, right, you can see that the Yorkie activity was quite a distance away from the tumor as well. But J&K activity is also found in fully attached cells, partially detached cells, but also fully detached cells as well. And unpairs are only found in detached cells, whether they were partially detached or fully detached. So this led us to believe that as tumors grew, they detached cells. This detachment leads to the activation of J&K and Yorkie, and the production of unpairs, which then were harnessed by the tumor to grow. To show that detachment can actually activate stress, I overexpressed metallomatrix proteases for just a few hours, like 6 hours, and you can see that the epithelium just sloughs off the basement membrane. And in those cells, you'll see activation of J&K and also unpairs. Another type of experiment you can do is you knock down integrins over time, and you see that the cells start to detach from the basement membrane and activate J&K, compared to control midguts, where all the basement membrane components are still, the integrins are still there. You can see that the depletion of integrins can lead to a regenerative response. There are mitoses, so stem cells are dividing to regenerate the tissue because these cells are detaching and probably on their way to be lost. And if you produce tumors that are surrounded by cells where they are progressively losing integrins, this actually allows tumors to expand further and grow. So this tells us that tumors can detach cells, and these detached cells can activate Jun N-terminal kinase and deactivate Hippo and activate Yorkie. This leads to the production of unpairs, which is then hijacked by the tumor to grow out further. Now, I only told you about this part of the story. The other part, there's a whole other part of the story, which I didn't get to tell you about, but I'll just run through it in this diagram. So upon the loss of differentiation, you form a tumorous stem cell. And what we found is that this tumor stem cell can reside in the tissue and not form a tumor unless it receives a signal to divide. And that signal is likely from tissue stress. Because if you provide the tissue with stress, they will divide and form a tumor. If you accelerate its cell proliferation, it will form a tumor. But once the tumor is initiated, it will initially grow out using autonomous EGF signals. But once it reaches its critical size, as I showed you, it will induce this niche, very reminiscent of a regenerative niche. And this will lead to these tumors to co-opt the niche as well. Now, interestingly, because of this regenerative environment is formed around the niche, other tumorous stem cells can actually start off forming a tumor because they're receiving this positive feedback as well. Now, many other types of tumor, fly gut tumors, especially, for example, the loss of APC or the loss of BMP, these are genes that are lost in our guts in colorectal cancer, can also form a regenerative niche. They activate J&K as well. And so the question that we have now is that since we know that damage, and these cells look like damaged cells, can activate reactive oxygen species production by NADPH oxidases, what role do NADPH oxidases have in this process? So could the cells actually activate reactive oxygen species production around these tumors, and that is causing these signals to be activated, such as J&K and unpairs, unpairs, but also the deactivation of Hippo signaling. Okay, so with that, I'd just like to conclude by thank you all for listening, but also thanking my current team members, my former team members. I have to thank the Wellcome Trust and the Royal Society for their generous support of our lab. And I have to thank Bruce, Edgar, and Aurelio Telemann, who supported me to do those 2 stories that I told you about, or finish those 2 stories, and all the people in the Edgar lab who helped with those, such as Devanjali Dutta, and who worked with me on the tumor project, which is the last project I discussed. Chloe Penalva, Michal Kardov, Bojana Pavlovich, Marian Roca, and Anya Thiel, who worked on the NOx and P38 story with me. And if you want to find out more about our research, you can go to our website, gutstresslab.org. And we also have a Twitter, so you can follow us on Twitter. And I'm currently also looking for a postdoctoral research associate. So if anyone has a strong interest in stem cells and tissue regeneration, get in touch with me. Thank you.
So thank you, Parthi, for the wonderful talk. Are there any questions? Yes.
Thank you. Good question. Very good talk. Very interesting.
Thank you.
I would say comprehensive. Doesn't seem to work.
Oh, no.
I will use it then. So
Does this mean I can unmute myself?
the second part where you were mentioning not kinase one, paper of the signal regulating kinase, this K1. So
Right.
it's really well conserved for evolution, and it's present in flies, and if you compare it with fly, insect, and mammalian, it will be all.
Mm-hmm.
But and also the mechanism. We were talking about our so in mammalian tissues, cells, the pathway would be to oxidize thyroidoxin or glutathione,
Mm-hmm.
which acts as a physiological inhibitor
Mm-hmm.
of the SK1.
Mm-hmm.
So what about flies? How this system is designed in that? Is it the same or is it something different?
Right. So actually, I think at least in the regenerative context, we don't know. So that's one thing I wanted to do was to overexpress thyroidoxin and see if we can inhibit ASK, because the model is that thyroidoxin inhibits ASK, and then you could maybe perhaps block. So there could be a negative feedback loop that regulates.
Yeah, it would be difficult because you don't know what to study.
Yes.
One would be it is phosphorylated already.
Right. So there's a paper.
Thyroidoxin won't
Right.
it just needs to be in sort of non-phosphorylated form.
Well, there is a paper from there's a series of papers from this group that actually argues that oxidation happens after phosphorylation.
Oh.
And this oxidation is of these different cysteines is important for the oligomerization of ASK, which is important for its activity. So if they remove all the cysteines, they still see that it's phosphorylated and activated, but it really cannot activate J and K signaling, for example, without those cysteines being oxidized, and it won't form these oligomers without those cysteines being oxidized.
All right. Thank you.
Yeah.
Any other questions? Yes, Maria.
Yeah, great talk. I just on the list and I
Thank you.
I was wondering if you had investigated whether these tumor cells can metastasize and form their own niche.
Right.
Wherever they relocate.
Yeah. So
Especially when they detach, if I may say,
Yeah.
like, when you for example, interfere with integrin and any
Right.
extracellular matrix proteins.
Yeah, so I did those experiments. The problem, that experiment is very difficult. So the MMP experiment and the integrin experiment I did with normal epithelial cells, and those can detach and they can activate these pathways. But the stem cell is very sensitive to it. If you lose the stem cell, if you overexpress RNAi towards integrins, or if you, matrix metalloproteases, if you overexpress them. So that was one thing that I had tried to see if that could be a way to, I don't know, manipulate the tumor mass. So that experiment is quite difficult to do. So what you need is a system where you can make a tumor and then turn on these things. Because I wasn't able to do it by just combining let's say, the gene that I was interested in manipulating and then creating a tumor at the same time. Yeah.
Any other questions? Yes, Susanna.
I've got questions. I'm getting preface this first, and I'm not a biologist, so obviously this is sounds really stupid. Okay.
Mhm.
I was quite interested in what you were saying about the tumor cells and the massive tumor. Do you ever get the case either in flies or maybe in humans? We actually generate tumor cells, and it's kind of sloughed off. You just excrete it, you get rid of it without actually realizing that there is actually a massive tumor cell.
So you're saying that the tumor cells actually,
understood you correctly, you've got tumor cells that kind of covered up with your normal cells. And if you
Oh, yes,
take that and you can break them into space and they expand.
yes,
Whether they kind of slough off.
they
And then
do.
it's excreted, but you don't actually ever notice that the problem carrying the tumor.
Right. So you only notice it when you do, well, I only notice when you start doing confocal stats, when you're looking actually at the tissue, it's not so obvious, and then you go through a confocal stat and you find various examples. I hope I understood this correctly, but you'll find a tumor mass that is completely covered by epithelial cells, and you'll find ones that don't have that. So I believe that they probably are stressed. They are eventually being lost and sloughing off and almost like leaving the tumor open.
But then does that then kind of regenerate the tumor mass will grow, or does it just get rid of it and then you reform with normal cells?
Oh, on top of it. No, we never saw that. But of course, maybe we didn't look long enough, right? But I don't, probably not. Because I have a feeling that, well, we don't know what the tumors are secreting, but you probably would have to secrete something that would be a good basement membrane apically that would allow for cells to crawl over or replenish that area. Yeah.
I actually have a question,
Yeah.
which is a little bit more general. So you showed us how ROS affects the proliferation and regeneration, and you also talked about tumors. And I was wondering, also if we compare to ourselves, right? When the gut is affected, how this affects the overall fitness or the lifespan or the survival of the flies.
Sorry, when what is infected?
So if you have, for example, tumors
Yes.
in the fly gut,
Right,
does
yes.
this affect the fitness of
Oh,
the
yes.
individuals or the lifespan?
Yes. So okay. So our system was a bit artificial. So we created tumors all throughout the gut, and those flies do not survive. They do have a shortened lifespan. But flies actually, I made this artificial, but over time, so other groups have been looking at notch tumors as well, and Alison Barden's group at the Curie actually found that these same tumors form normally within flies, particularly males. So over time, with age, tumors do form in the gut.
So that would mimic sort of what happens if you sort of insert some.
Exactly.
Okay, great.
Yes,
It was related to what Tanya just asked. So if you look at other parts of the fly organs, like the brain, after stressing the gut.
The brain, not yet, but that's a great question. Yeah, we've been interested in that.
We know there is a connection in the last years.
Yes.
Things like Parkinson's disease starting in
Right.
the gut. So
There's also a lot of connection between the microbiome in the gut
yes,
and the brain.
exactly. They found that, I think it's a recent study published in 2021. So, just.
Yeah, no, that's
what
something,
I've done
it's funny you
and-
mentioned that. That's been on my mind. Yeah. So many things to do.
Great. I don't think, do we have any questions on the chat? Yes, okay.
It's Jerome.
Yes, Jerome, please unmute yourself.
Hi, Parthiv. Really interesting talk. I was wondering about the ROS part of your talk. So,
Hey, Jerome. Can you hear me?
Can you hear me?
Yes.
Yeah, so I was wondering about the ROS part of your talk. As you know, in aging, it's also, there's still the ROS theory of aging. Have you looked at any of these ROS components, and if kind of like either mildly overstressing them, or so mildly increasing ROS or mildly suppressing ROS production in ECs can actually extend lifespan?
No, we haven't done that. No, but that's a very interesting thing to look at.
Yeah, it would be kind of cool to know, like, especially in this, ROS driven theory of aging that kind of went out of fashion, but it's now, there's some new studies out that kind of say again that indeed excess ROS production can indeed cause age-related problems, right? So
There's also the opposite. So I'm not sure if you know about this paper from Linda Partridge's group, where they did redox proteomics and found that flies that are aging actually don't have accumulated oxidation.
Okay, yeah.
But they do upon fasting.
Yeah, Yeah,
So
that's
It's like the opposite of what you would expect.
exactly. So yeah, no, it's I think it's an interesting field to look into. But yeah.
Yeah, absolutely.
Good. Okay, thanks.
Mhm.
Great. Is there any other? No. That was it, right? We don't have any other questions? Okay, great. Then we can finish today's seminar. Parthiv, thank you very much
Yeah, you're welcome.
for coming here. Thank you very much for your attention and coming up here today. And have a great day. I think we can